So in this model here, we will be representing muscle contraction, and this is our rigor mortis model. So here we have the dark purple pipe cleaners, which are representing our Z line. The straws here are representing the myosin filament. The white beads represent the cross bridges. The light purple pipe cleaners are representing the troponin, and then the blue pipe cleaners right here represent the tropomyosin. So another thing that is active during muscle contraction is ATP, and that what it does is it helps the muscle return to its relaxed state.
As you can see in this model, the myosin myofilament is represented by the red and white straws. It is composed of primarily the protein myosin. Each straw is a filament, and each filament has more than 100 molecules of myosin. The white beads represent the myosin myofilaments, which interact with actin myofilaments. During muscle contraction, the axon terminal of a motor neuron will release a neurotransmitter.
The neurotransmitter, after being released, diffuses across the synaptic cleft and binds to muscle fiber. Impulses travel to the sarcoplasmic reticulum. At this point, the sarcomere is at rest, which means there will be minimal interaction between the filaments. However, calciums are released from the sarcoplasmic reticulum into the sarcoplasm, which binds to troponin. After this calcium binding, troponin and tropomyosin undergo a change, which exposes the myosin binding cells on actin, or the thin filaments. The thin and thick filaments then link together. After binding, the thick filaments pull thin filaments toward each other inward. Rest and contraction. When these muscle contractions occur, the sarcomere is shortened. If there are multiple sarcomeres lined up to each other, they would be standing next to each other back to back, and that is muscle contraction in the sarcomere.